Imagine being able to separate out the individual colours in a pen, to uncover the chemicals in your food, or even to help the police identify substances at a crime scene. That's the power of chromatography. It's a simple but brilliant technique that lets us split complex mixtures into their individual parts. In the lab, it's used to check the purity of medicines, analyse dyes in fabrics, and even test for the misuse of drugs in sport. In this episode, I'll show you how chromatography works, why it's so useful, and how you can master it for your GCSE Chemistry course. Welcome to the fourth and final part of our separation technique series. There's an overview available, which is linked above, and the channel has separate videos available on the details of each technique. Today, however, we're looking at chromatography, and I'm going to show you how it can be used as an analytical tool. Chromatography works on the basis of the relative solubility of various chemicals in a solvent compared with their attraction to the surface of a substrate. Right, there's quite a bit of language there, and to be honest, a fair bit of theory to get your head around. So let's start with the key terms. We'll start with a mixture of chemicals. For example, those contained in the black ink of this felt tip pen. We'll call them the components of that mixture. Now, if I place a dot of the ink here on this bit of paper, then those components are currently fixed in one location. But by adding a drop of water, as it spreads out, it carries the components of that ink away with it. The paper itself didn't move, so we're going to call that our stationary phase. The water did move, it moved through the paper, and so we're going to call that our mobile phase. You might notice that the components in the ink didn't move quite as fast through the paper as our solvent did. And that's because they were pulled down into the fibres of the paper a little bit, and so they were kind of sticking to the stationary phase. Now, there's actually quite a bit of clever chemistry going on there to do with water layers on cellulose fibres and a process called adsorption, but that's for A-level and beyond. We can just think of those component molecules getting pulled down onto the stationary phase and held a little bit as they pass by. The second thing you'll notice is that some of the components got slowed down a bit more than others. And this means that it was rather like watching a group of athletes with different speeds running a race. And we can use this technique to split a sample up based on the relative movement rates of those components. Let's take a look at that race metaphor a bit further, because it's really helpful here. Now, rather than starting at the middle of a bit of paper, let's create a track with a start line. This is a strip of chromatography paper, which is specially made to have a consistent structure and a known chemical nature. It's important that my start line isn't going to move, and so I'm going to draw it with a pencil so that it won't dissolve and move up my paper with my sample. Next, I'll place my ink dot here, right on the start line. And then I'm going to hang this bit of paper on this support so that the very bottom of it will dip into my chosen solvent, which in this case is water. I'm being very careful to make sure that the ink dot itself doesn't get into the solvent, because otherwise it will just dissolve away into the whole liquid base at the bottom, and that's the last thing that I want to happen. My water, that is my solvent, my mobile phase, now moves up the paper. It dissolves the components as it reaches the ink spot and begins to carry them up the paper with it. All of the components are attracted to the stationary phase to some extent on the way up, and so they all hang behind the leading edge of that solvent by various amounts. We'll call that leading edge of the solvent the solvent front. And actually this is a lot like looking at a racetrack from above. And just like a race, if I leave it long enough, all of those components will reach the end of the track. They will no longer be spread out. What I want to do is get a snapshot of the race when the components are as spread out as possible. And that will be when the fastest of those components, that is the most soluble in the solvent, has got nearly, but not quite, to the top of the stationary phase. By removing the paper from the solvent at this point, the solvent front stops moving and I get a freeze frame of the positions of my components. Now imagine if we could look at this bit of chromatography paper from the side as the solvent moves up it. It might look a bit like this. There's the components of the ink, all in one place at the start. And then, as the solvent passes them, each one moves at a different rate due to their relative solubility and their interactions with the surface. The blue component, being the most soluble in the mobile phase, travels the furthest, while the yellow component, being the least soluble and with the greatest attraction to the stationary phase, moves the least. And that's it. You know the theory of chromatography. I'm now going to show you how to carry out a chromatography experiment in a reliable and repeatable way, as this is a skill all chemists should know. And it's also one of the required practical skills for a GCSE in chemistry in the UK. To make this procedure really clear for you, I've prepared some little animated diagrams to support the explanation, and I'll be using these along with performing the experiment live in front of you so you can see how the practical is actually carried out. So the preparation for a good chromatography experiment starts a significant time before the paper hits the water. The first step is to prepare the mobile phase, and we do this by adding a centimetre or so depth of the chosen solvent to the bottom of our beaker. Then we place a lid over the top of the beaker and leave it for at least 10 minutes. This will allow the volume of air inside the beaker to become saturated with a vaporised solvent. This will prevent or at least reduce the evaporation of the solvent from our chromatography paper during the experiment. This isn't something that most chromatography practicals at GCSE mention, but it is an essential part of getting a good, high-quality, reproducible set of results when chromatography is done in a professional lab. Now we move on to prepare our chromatography paper that will be our stationary phase. Assuming that you've already been given a pre-cut length, we'll measure about 2 centimetres from the bottom, and then we'll draw a line with a sharp pencil across the paper. This will be the line upon which we place our samples, and it will be the baseline that we use to measure the movement of both the solvent front and the samples at the end of our experiment. We then place a small dot of each of our samples at a spacing of at least 1 centimetre apart along that pencil line. Touch the paper as lightly as you can here, because chromatography paper is very delicate and we don't want to damage it. I should be able to spot the difference between these, but you never know. So I'm going to label them, as I said before, with pencil A, B, C and D, so that I know what my samples are later. Because actually, once those colours split up, they may not be easily identifiable anymore. Now, I'm using the felt tip pens, but you might be asked to prepare a chromatography paper using a liquid such as a food colouring. Now, all I've got here is just a little white tray, little sample tray with a tiny dot of one of the food colourings here. And I'll just show you how I'd prepare that. So what I've got is I've got a little glass capillary tube here, which is a very thin-walled, very fragile tube with a tiny hole in the middle, through the middle of it, about 1 millimetre thick. I'm just going to touch that on there, and capillary action will just pull a little bit of that dye up into my tube, and then all I do is lightly touch my tube very, very briefly onto the bottom of my paper, like that, and I get another little spot of colour just there. We should then fold the top of the paper over a support rail of some description, which is usually a glass rod or a wooden splint. And then we need to pre-check the length of our paper to make sure that it does not touch the bottom of our beaker and that the pencil line will be above the level of the solvent in our beaker. Once we've confirmed this and adjusted the length of paper if necessary, then we clamp the paper in place with a paper clip, remove the lid from the beaker and carefully lower the sample into the beaker so that it hangs straight and level without touching the sides or the bottom. We replace the lid to ensure that we keep saturated air inside, and then we wait while the solvent makes its way up the paper. Once the solvent is a couple of centimetres or so away from the top of the paper, we remove the lid, take out the sample on its support, and before we go any further, we should mark the height at which the solvent reached, which is something we're going to call the final solvent front in our calculations. And then I can look at my 4 samples. Sample A contained 1, 2, 3 clear components. Sample B has 2, and samples C and D seem to have only 1 clear component each. We now enter the analysis phase of the experiment, and we're going to measure something that we call the retention factor, or the RF value of each component. This is the relative distance it travelled up the paper compared to the solvent front. Now, this is where real life is a bit different to the exam. As you will already have seen, my spots have turned into kind of messy blobs really as they've gone up the paper. And this is normal when using chromatography paper. However, in the exam, you'd normally get something printed out that looked like this. Because our next step is to measure the distance from the starting baseline to the centre of each spot of colour. Now, using a printed example like this one, that's pretty straightforward, as you just measure from the baseline to the centre of each circular spot. Dealing with these much more spread out areas of colour from my actual sample, I first need to make a judgment of where the centre of each spot is, which I then mark with a little line, and then I use my ruler to measure from the baseline to that centre mark on each spot in turn. You will need to present your results in a neat results table. However, my tip for you is to only draw the results table once you've seen how many components there are in each of your samples. Having seen my results, I drew this results table into which I was able to copy my results very neatly. With those values now in my table, we're ready to calculate those all-important RF values for our samples. The RF value is just a fraction, with the distance moved by the component at the top and the distance moved by the solvent, that's the distance from the baseline to the final position of the solvent front, on the bottom. Remember to check that your units are consistent, and then it's time to reach for the calculator and convert all those values into decimals. These are normally given to 2 decimal places. Before we look a bit closer at the results, notice that there are no units for the RF value. That's because it's just a ratio of the distances travelled. By the way, the RF value for a chemical should be consistent for the same solvent and same substrate combination. So, if my RF values are the same, then I could be looking at the same substance present in different inks. 3 of my inks all had a blue colour in them, and those components all have very similar RF values, certainly within the tolerances of my experiment. And so there's a good chance that they are actually all made from the same chemical pigment. The orange component in ink A looks similar to the orange ink in ink D, but this time their RF values are not close enough. So either it's a different chemical, or there's another component in ink D that's overlapping with it. I would need to repeat experiment D with another solvent to see if that was the case. And that's about as far as we can take things at GCSE. But chromatography has quite a few more tricks up its sleeve when it's used in a professional laboratory. At GCSE, you'll probably only look at water as your solvent or mobile phase. But bear in mind that you could use any solvent. Some chemicals simply won't dissolve in water, but they will dissolve in perhaps ethanol. Secondly, no professional lab will use chromatography paper as their stationary phase. It's a little bit inconsistent, and the components' interactions with the stationary phase are a little bit tricky to predict. Instead, they use this stuff, which is a thin layer chromatography plate. Here, the stationary phase is a very tightly controlled film of silica or aluminium oxide particles, each around 10 micrometers across. And this will give results much more similar to the diagrams you get given in exams. But these plates cost around 10 times as much as the paper option, and so you can see why you probably won't meet these until A-level. Finally, I'd just like to point out that we've been looking at analytical chromatography, which is where we split up very small samples to see what they contain. Chemists use a similar process to separate larger volumes of mixtures using columns of silica powder, or machines such as this, which perform a similar job semi-automatically. It's machines like these that are generally used by forensics agencies and pharmaceutical companies because of their high accuracy and reproducibility. But you're unlikely to see one close up until you're at university or working in an industrial laboratory. In summary, chromatography is a versatile tool that can split samples based on their relative solubility in a solvent compared to their attraction to a given substrate. For a GCSE exam, you should be familiar with the key terms solvent, substrate, component, stationary phase, and mobile phase. You should be able to calculate an RF value from either tabulated data or from an example chromatogram that you were given. To help you with this, there's a worksheet available on my Patreon page, which is free as always. And if you need a bit of help, drop a comment below and I'll get back to you with an answer. Otherwise, thank you for watching, and please drop me a like and subscribe to the channel to get more content. See you next time.